Hello students, this is Brian. Welcome to Biopsychology Exam 2 Online Review Session Part 2. I will go over Chapter 5 on this segment. Note that I will only go over what is important in the slides. You are responsible to know most of the material in the lecture notes, but I will pinpoint the more important ones. This online review session is the recap of my in-class review session. Alright, let's continue. So here are the examples of the visual imaging techniques, and then for the structural brain imaging technique, we have contrast X-rays, CT scans, and MRIs. For functional imaging, we have PET scans and fMRIs. Keep in mind that functional imaging measures activity. So you need to know contrast X-rays, and you need to know its method, and you need to know an example, and I've highlighted it on the slide. And then here is the computed tomography. You need to know that in exam 2. And then what we know about this imaging technique is that it yields 3D representation of the brain, and you need to know its method and how it works. And then now we're talking about the magnetic resonance imaging, which is the MRI. So basically, this is a high-resolution images constructed of measurements of waves that hydrogen atoms emit when activated by radio frequency waves in a magnetic field. And then what we know about MRIs is that it does not require injection of substance. It uses a magnet and aligns your hydrogen atoms based on your magnetic poles. Then a radio wave is applied so that it spins simultaneously. And then now we're talking about the PET, and you need to know its method. And then basically, you inject 2D. You inject the deoxyglucose into the carotid artery, and then 2RD is taken up by active brain cells but not metabolized. So what ends up happening is that it yields a colored map of radioactivity that can be superimposed on brain image. And then here's an example of what it looks like. And then now we're moving into the functional imaging techniques. And then now we're talking about the fMRI. And what it does is that it looks on images of areas with increased oxygen. And then it works, and then we're wondering why it works. It works because active areas of the brain takes up more oxygen than they need, and oxygenated blood has magnetic properties. The measurement that it uses is the Bolt signal, which is the blood oxygen level dependent signal. We need to know this for the test. So even though visual imaging techniques find interesting things, it doesn't change that they are all correlational studies. So to find causation, we need to find a way to stimulate the brain. So one technique that allows us to do that is the transcranial magnetic stimulation. So what it does is that it makes yourself fire by using magnetic stimulation. It makes yourself fire continuously until it reaches a fatigue stage. And then basically it disrupts the brain region under the skull. And then keep in mind that we use this to assess how behavior or cognition is affected. So it creates a sham, it's a control or placebo group. And then here are this is the outline of the psychophysiological activity, which is the EEG, ERPs, muscle tension, eye movement, skin inductance, and cardiovascular activities. You all need to know this for an exam. So basically, the EEG measures the gross of electrical activity of the brain, and these are different types of waves: aroused, relaxed, asleep, and deep sleep. You may have to notice from the test since we talked about it in class. And then you need to read this part over here because it's important. I'm just gonna highlight for you guys. And then now we're talking about the event-related potentials, which are the ERPs. So basically, these are EEG waves that accompany certain psychological events. And you definitely need to know sensory evoked potentials. So it's actually change in EEG elicited by the presentation of the stimulus. And then basically what happens is that the wave is averaged over several trials. Each wave is characterized by direction and latency. And this figure is an example. And here's my side note from the lecture. So now we're looking for muscle tension, and it's called electromyography (EMG). So basically, what happens is that increased muscle tension equals increased amplitude of raw EMG signals, and total EMG is equals smooth out form of raw EMG. And here's the side note. You need to know this from the exam as well. So now we're looking at electrooculography, which is EOG. And then here's my side note also. Pause the video if you want to look at it. And then basically, you need to read this part over here. And you can see the retina is negative and the cornea is positive. And then when it moves, you can record a change in electrical potential. And then now we're looking at skin conductance. So basically, skin conductance level is the measure of the background level of skin conductance that is associated with a particular situation. And skin conductance response is measure of transient changes in skin conductance that are associated with discrete experiences. So these are the things that makes these two different, and you need to know this for the test. And then now we're talking about the cardiovascular activity. So then we talked more about the blood volume, which is the measure of how blood is present in a particular part of the body. And then here is my side note. And now we're moving into invasive physiological activities. So basically, invasive means we insert stuff in the brain. So one invasive physiological activity is the stereotactic surgery. So you use this to position experimental devices within the brain, and then we also use a stereoactive atlas, and it provides coordination for locating structures within the brain. And we also use the stereotactic instrument, so it's used to hold steady and guide the device, to be inserted. And here's my side note as well. So then, now we're talking about lesion. Lesion is to remove, damage, or destroy part of the brain to observe impact on behavior. And then you need to know these different kinds of lesions. And then one key point about lesions is that it must be interpreted carefully because it is difficult to make small, precise lesions in the brain. And then here is my side note as well. And then now we're talking about electrical stimulation. So basically, what it does is that electrical stimulation may be used to activate a structure. So what it does is that stimulation of a structure may have an effect opposite to that seen when the structure is lesioned. Here's my side note. So now we're going into invasive electrophysiological recordings. And you need to know these four: intracellular unit recording, extracellular unit recording, multiple unit recording, and an invasive EEG recording. And here's my side notes. Pause the video if you would like to write this down. in this slide, you need to know and you need to be able to identify which type of recording is this, and you'll be asked in an exam to identify which of one is which. And now we're moving into pharmacological research methods. And then here's the outline of what you need to know for an exam. So the routes for drug administration, selected chemical lesions, measuring chemical activity in the brain, and locating neurotransmitters and receptors. And then here though, this is and then we all know that it must pass through the blood-brain barrier. And then these are like the methods of drug administrations. You need to know them. I think that you need to know them. All of these. And then if it cannot pass the blood-brain barrier, you would use a cannula. Cannula is a fine hollow tube that has been stereotaxically implanted in the brain. And then now we're talking about selective chemical lesions, and it is important to know neurotoxins. That's to be an exam. And here's my side note. Pause the video if you would like to write this down. And then now we're moving into measuring chemical activities of the brain. So then we're using a 2-deoxyglucose, the 2DG technique. And then you need to know this in the exam. You really need to know this in the exam. So you read this entire slide. You also need to understand cerebral dialysis, and then basically what it is that it measures extracellular concentration of specific chemicals in live animals. And here are the characteristics, and you need to know this for the exam. Now we're moving into pharmacological research methods in which we locate neurotransmitters and receptors. So basically, there are two ways to do it. This one is immunohistochemistry. It's basically based on binding of labeled proteins, protein-specific antibodies. So basically, it looks for immune response. Antibodies created that bind and destroy antigens. And in situ hybridization, it uses labeled RNA to locate neurons with complementary RNA. And here's my side note. Now we're going into genetic engineering. So you need to know the gene knockout techniques and the gene replacement techniques. You need this for an exam. And then now we're moving into cognitive neuroscience techniques. So you need to understand the assumptions and the goals of the neuroscience techniques. And then here is the paired image subtraction. So basically what it does is that it produces different images between two tasks that differ in a single continuous cognitive processes. So basically what it does is that it signal average so what it does is that we would subtract hold on. Yeah, you look over here. So here's an example of my side note gives an example of how paired image subtraction works. Sorry about that, I'm running out of time. Important to note that that paired image subtraction uses signal averaging. So basically, it averages the different images across subjects. What it does is that it controls for random events. And this side note, I'm going to bring it up again, and then I'll try to be more clear. Is that this paints you this example gives you an idea on how paired subtraction works. And now we're moving into behavioral paradigms for lab species. So the first thing we're talking about is species common behaviors. So basically, what we know about is that behavior displayed by virtually all members of the species. And you see them grooming, swimming, eating, drinking, all that stuff. And then there are three ways to test common behaviors. One is open field test. Two is test for aggression, defensive behavior, and test for sexual behavior. So basically, you need to know the open field test, and then basically you need to know its measurements, and what it does. So the open field test, what it does is that it measures fearfulness, and you measure it by automated activity recorder. You would draw lines on floor and count the number of crosses, and then the number of boluses. Another way to test it is to test it for aggressive behavior. So basically, the method you're using is a colony intruder paradigm, and it measures aggressive behavior by alpha male and defensive behavior by intruder. And then this side note shows the characteristics of attacker and the defender, which is the intruder rand. And another thing that they test common behavior is the test of sexual behavior. And then there are three measures: the amounts of intromission, and the number of intermissions to ejaculation, and intervals between ejaculation and the next mount. And then now we're talking about traditional conditioning procedures. One of them is the Pavlovian conditioning, and this is going to be an exam. Here's my side note. And then upper conditioning, it's reinforcement and punishments. And then here's my side note. And then sub-stimulation, which is an animal works for electrical stimulation. And here's my side note. And I'm going to make a little comment about sub-stimulation. It seems to be more satisfying for rat than eating food. So basically, when they stimulate the brain that is responsible for the reward system, the rat will continue pressing it. And then basically what happens is that if you find a correct stimulation, what happens is that the rat will continue to press button and die from starvation even though the food is present. And now we're learning a semi-natural learning paradigms. You need to know conditioned taste aversion. You need to know the radial arm maze. And you need to know conditioned defense burying. And then for wrapping it up, you need to know convergent operations. So basically what it does is that, one strength of one method balances out the weaknesses of others. And that's it's important. Alright, and this concludes this concludes exam 2 online review session. I'm hoping that this will help you study for an exam, and I'm going to remind you that the exam will consist. Some of the questions will be from the book. It's about 10%, and 90% of it will be from your lectures. And then, even though I didn't say a lot of this is going to be an exam, pretty much the things that I mentioned are going to be an exam. But I made special notes on some things you need to pay attention. Alright then, happy studying, and I hope you guys get good grades. Thank you for watching, and please, if you have a YouTube account, make a comment, and then if you think that I need to work on something or make improvements on my next review session, please do so. I need the feedback. I need all the feedback from you guys so I can see what I can do to improve this presentation. My goal is to help you guys do well in the class and learn, not just really about the grades, but I would like for you guys to learn about biopsychology. biopsychology is the future, of psychology, and I hope you spend your weekend working on it, studying for an exam, because I know the exam is not easy, and then I'm hoping that this study guide works and it helps you. All right, have a good day.